thank you, everyone. Thank you, everyone, for coming. Thank you, Sophia and Andrea, for organizing this wonderful session. I've had a great time. I can't see the slides on my computer, so I'm going to be sort of running back and forth. So I'll be telling you a little story about a little pet hypothesis that I've been working on for many, many years called Mother's Curse. So that change?
Do you want this?
Yeah, let's do that. Excellent. Everyone, excellent. So we're all gathered here today because we all love mitochondria, right? So I don't have to talk too much about why mitochondria is extremely important. But mitochondria produces all the energy. It does a lot more. But the main dogma is that mitochondria produces all the energy that we need to survive. Mitochondria has its own genome, which we call the mitochondrial genome. And even though this genome is small in size, it's 17 kb in flies, it's extremely important in evolutionary processes and physiological processes as well. And there is a strong support for coevolution, as we saw in this morning's session, between the mitochondria and the nuclear genome. So I really like this slide. So if we zoom into the mitochondria, here we have the cartoon diagram of the electron transport chain. We have several different complexes. Complex 2 is missing, so I'm sorry, complex 2. But what we can see is that it is composed of several subunits. Each complex is composed of several subunits that come in two different colors. So the ones in blue here, these are encoded in the mitochondrial genome, and the ones in yellow, they are encoded in the nucleus. So I really like to show this slide because you can really visually see that how important this interaction, its physical interaction between these two genomes are. And many people in this room, including myself, have spent a lot of time trying to study what happens when things go wrong, either via mutations in the mitochondria or in the nuclear genome. Now things get a little bit more complicated because the mitochondria is inherited maternally, right? And so if we think of selection as a sieve, and in the mitochondrial case, a female-specific sieve, where all the new mutations that arise in the population must interact with the sieve. The mutations that are good for females will pass through the sieve, and mutations that are detrimental for the females will get stuck in the sieve and removed through strong purifying selection. Now the question arises whether this form of selection will benefit both males and females. In theory, it could be. But in the 90s, Frankenhurst published a really important paper to me, sort of highlighting the fact that this form of selection could select for sexually antagonistic mutations. So a mutation that is good for females, but bad for males, right? And these would get through the sieve and passed on to the next generation. And, you know, we could think about, would we expect lots of these sexually antagonistic mutations to go through the sieve? And because the mitochondria is heavily involved in metabolic dependent traits, metabolism itself, it's a sexually dimorphic trait. So we could predict that a lot of the mutations that benefit female metabolic rate, physiology, or traits that are highly underpinned by metabolic demands might be sexually antagonistic. And as any evolutionary biologist, we look at things through time. And through time, we expect the mitochondrial genome to accumulate a lot of these mutations that are male harming, but female beneficial. And I'm not the only person thinking about these things. In 2004, I believe, Neil Gemmell and colleagues wrote this really nice review, sort of coining this process. He gave it the flashy name of Mother's Curse. And the story behind it, it's mothers are cursing their sons with bad mitochondria, or mitochondria that has accumulated a lot of mutations. Right. So when I first joined the lab, so in Australia, did my PhD in Australia, in Damien Dowling's lab. I was a very naive PhD student, master student actually, and Damien just said, you need to do an aging assay, because aging is a really good trait to look for signatures of mother's curse, because it's sexually dimorphic and it's heavily underpinned by metabolic demands. So I naively undertook this horrible aging experiment, which I'm so happy that it gave really good data. So in this work, from about a decade ago now, I feel really old, we have, down here on the X, we have lots of different mitochondrial haplotypes. These are all fly genotypes. All the fly genotypes have the same nuclear background, and the only thing that differs between these genotypes is the mitochondrial DNA. So we have mitochondria from all over the world in this panel: Alstonville, Barcelona, Brownsville in Texas, so on, Sweden, and Zimbabwe. Right, so this panel was really cool, and it proved to give a lot of support for mother's curse in a whole bunch of different traits that we measured. And here, the main thing that I'd like you to get across is that to look for the signatures of mother's curse, we want to see that the mitochondria is not having much of an effect, except for this guy over here, girl over here, in females. But when you change the mitochondrial genome, you get a lot of variation in the phenotype in the males. And in a separate paper, I was able to get some evidence that this might be driven, this difference in variation might be driven by differences in expression of complex 1 genes. One of the issues that we had from this work, which I raise my hand, guilty, is that it's not generalizable, right? We look at one isogenic nuclear genome, whole bunch of different mitochondria. How can we make the story of mother's curse more generalizable? And so several years ago, I think the summer before COVID lockdown, what we did is that we tried to create a new panel where we could test these ideas using a lot more nuclear genetic variation. And so for this, we looked at the Global Diversity Panel of flies. This is flies that were created by Andy Clark's lab at Cornell. They basically got flies from all these different places in the world. They created between 20 to 30 isogenic lines, and then they sequenced everything. So there's a panel that Andy has about, let's say, 150 lines. Everything's sequenced, everything's inbred, and it's really good to do sort of genotype to phenotype studies. And so we took advantage of this. We selected 9 out of these 100 and something flies, and we basically targeted fly lines that had quite diverged mitochondria and nuclear genomes so that we could maximize our chances of finding different things. And what we ended up doing is we created that summer a big panel of flies where we coupled every possible nuclear genome to every possible mitochondrial haplotype. And I was going to sort of say it's the biggest panel, but took who's somewhere around here. You have 300 and something. So kudos, chapeau, well done. I can't say that anymore. So I can say it's the second biggest mitonuclear panel that I know of. So we created these 81 genotypes, and it's a really nice panel. Here you have all the total SNP differences between the mitochondrial genomes, so you can see the levels of variation. But it's a really nice panel to look at several different questions. Some of the questions that we look at is sort of co-evolved, which is the diagonal here, versus the mismatched, right? And we can also look at signatures of mother's curse, which is my little pet hypothesis. So I'll tell you this story sort of in chronological order as things happened. So as soon as we created this panel, we got this wonderful master's student, Lorcan, up here, which wanted to test mother's curse across this whole panel. Because it's Drosophila and it's a lot of genotypes, doing these really big phenotypic assays is really labor and time intensive, especially going into a pandemic. What we decided is that we would look at wings, because we know that wing is a sexually dimorphic trait, so tick on that prediction, and it correlates tightly with body size, big wing, big fly, and body size is a very plastic trait with heavy metabolic underpinnings. Right, so we thought that this would be a really good trait to measure, to look into mother's curse. And so Lorcan, we froze everything. He plated all the wings into little microscope slides, and then he measured a lot of these phenotypes at home with a microscope that we lent him. So he plucked lots of different wings. He measured everything, landmarked, and we calculated the centroid size, which is a relative area measurement. And just some data so that you can have a look. We did see the differences in the wing size. So males on the left are bigger than females, which is what we would predict. We also, if we group all the nuclear genomes, so if we group all the mitochondrial haplotypes per nuclear genome here, you can see that there is nuclear genetic variation for the trait, which is nice. So things are different. And then I'm going to show you the big plot, which has everything with everything. So here we have, each box is a nuclear genome, and down here we have the different mitochondrial haplotypes. It's quite hard to digest this plot, but in some nuclear genomes you can see that changing the mitochondria does change the wing size, and in some cases it doesn't. So what we look for when we look for testing mother's curse is actually look at the coefficient of variation. So how much variation is caused by the mitochondrial genome. So if we group all the nuclear genomes and look for mitochondrial genetic variation, for most of them, we find more mitochondrial genetic variation in males than in females, except for one. So it's not pervasive. Yes. Yes. But 8 out of 9 is still pretty good, right? So the conclusions from Lorcan's paper is, mother's curse could be more generalizable than we previously thought. So that was a really big thumbs up from me. I was really happy with these results. And since then, we've been really busy phenotyping this panel. So we have here a whole bunch of traits that we've measured. So there's a lot of work has been done on this. A lot of student postdoc and technician time has gone into this project. So I just want to thank everyone on this slide. But yeah, we've been really busy gathering all these different traits, which is pretty great. We've started, so this is again super preliminary data from this morning. We've started, yeah. We've started sort of doing this multivariate analysis with all the traits. So here I've just got all the different traits in a PCA, colored them by nuclear genome, and we can start seeing different nuclear genomes are doing different things in the parameter space. Here we have all the loadings. So for all the traits, we've measured several things. So we have a whole bunch of traits for both males and females. So this is a really exciting data set that we want to explore. But for mother's curse, the main thing, of course, is fitness. So we were able to measure the fitness data on time. And I just want to highlight these two great people in the lab, Annie McShane and Marion Bonneau, that each tackled these big fitness experiments across these 81 lines. So Annie did an egg-to-adult viability experiment where the flies laid eggs, we counted the number of eggs, then we counted the number of adults that came from those eggs, and then we calculated the viability. And Marion and the lab did, again, a really big male fitness experiment where we did a competitive fitness assay where we compared our males from the panel against other males for the mating of females, right? So that not only involves a little bit of sperm competition, but also male-to-male competition in our flies. And, oh, it's a bit blurry, so I'm really sorry. So this is so we bootstrapped the coefficients, the mitochondrial coefficients of variation. And again, here we see that across most of the nuclear genomes that we measure, the male mitochondrial coefficient of variation is greater than the female, except, again, for this pesky E genotype that we have to look at a bit more in depth. So it's there, it's not pervasive, but we see signatures of mother's curse, or we see some signatures of mother's curse. So the mtDNA has a greater impact on male size for all but one nuclear genome. So maybe mother's curse is kind of real, which makes me happy. I'm not betting on it yet, but we're getting some strong evidence. And so just to start summing things up for the future work, we really want to use a multivariate framework to incorporate all the different traits. So those PCAs that I've been showing you, we really want to incorporate all these traits into one big analysis that I have yet to know how to do. So I'm hoping any QuantGen people will love to do this work and collaborate. So this will allow us to fully test this hypothesis. So I really want to say, across all the traits that we've measured, we find signatures of mother's curse. And then I might be a little bit more convinced that mother's curse might be real. Some future work that we're currently doing in the lab is that not all genotypes are happy. We have 2 genotypes that are very unhappy. We have this genotype over here. If you have the nuclear genome F with the A mitochondria, the larvae don't develop. So there is something going wrong in instar, in the second instar, that they just won't go from second instar to third instar. They're dying, right? So we're trying to look for the molecular mechanisms. There has to be some really strong mitonuclear incompatibility there happening. So that's a really cool, more molecular project. And here, flies that have the G nuclear background with the I mitochondria. These genotypes are sad because the males are sterile. So I've done a couple of really preliminary analysis where I've dissected testes and looked at sperm coming out. The co-evolved, GG, you rip the seminal vesicle and all the sperm come out like spaghetti. For the GI, you rip the seminal vesicle and nothing comes out, right? We just need students and people to try and find the molecular mechanisms underpinning these 2 fly genotypes. And I think with that, I've got lots of time for questions. I just want to thank all the people that did all the phenotypes. I want to thank Stefano and Finley for being wonderful lab members, and Stefano's talk is on Tuesday in the other mitochondrial session. And I want to thank Kevin Max and Andy Clark's lab for all the help and support, and all of you for listening to my ramblings. Thank you. That is something that we want to do. So we've only done the divergence of mitochondrial SNPs, and we have the protein coding, the synonymous, non-synonymous ones. So that's something that we really want to do, look at the nucleus as well and try and find. Yes, George.
Great talk.
Thank you.
What's up with that 1 on 9 line that seems to be always telling you the same? That's always going against the pattern.
I have no idea, man. I have no idea. I mean, you know, we could look at the genome and we could look at the mitochondria, but you know, it's just it's the same one in both. So it's acting weird. We sequence them often. It's not contamination. We back cross them to their corresponding nuclear genome so that they don't accumulate nuclear mutations. It's always been like that. For most traits, E nuclear genome is a bit of a weirdo.
This might be a naive question.
No, go.
If you input the original mononuclear pair, do you think you'll be able to annul or reverse this difference that you're saying that's impaired or not really? Like as kind of a rescue experiment.
So, I mean, ideally, in ideal land, what I want to do, for example, for the FA, what we're currently doing in the lab is we're doing a sort of a mapping experiment where we're trying to map where the nuclear loci that interacts with the A mitochondria that is causing the incompatibility, and then we want to use CRISPR to CRISPR that bit to the A nucleus, bring it in, and then trying to see if we can rescue it. Yeah, thank you. Yes, Pierre. We haven't looked at aging. I'm sorry. We haven't looked at aging. That's a killer. That's a real killer. Thank you.
I was wondering, most of the time when we're looking at coadaptation or monitors, we're looking at animals that are living in a cozy environment after that, and where mitochondria we don't pass these mitochondria to their full capacity.
Yeah. Yeah.
Do you think that we miss some condition, or maybe it would be easier to reveal monitors in
Yeah.
stressful?
Yes. So I'm playing a lot with diet, and I'm in thinking about doing this evil experiment that I have to do because I said I would do it in my fellowship, where we put the whole panel on several different diets, and then we measure fitness. And so we will have a look at how diet stresses the mitochondria and how the different diets can shape the dynamics of the mother's curse that we find, at least in cozy 25 degrees, wonderful food. Yes.
First of all, this is great.
Thank you.
And this is a really heroic effort combining all these different genotypes. Just like a fun toy thought experiment.
Yeah, go for it.
You're talking about looking for reinforcing evidence for sport mother's curse. It's like a real phenomenon we see.
Yep.
And to your point, you're talking about how mitochondria exist in a lot of different environments that are outside the cozy lab conditions. Have you ever considered looking at this at like a population level rather than like the level of a given individual nucleotide, a given individual mitochondrial type, maybe introgressing a whole population so that you've got sort of natural, even like lab-specific population with one particular genotype?
Yes. So if you go to Stefano's talk on Tuesday, we did exactly that with two populations from the Australian east coast of Australia. So we have the north of Australia, which is subtropical, and the south, which is temperate. And I don't want to spoil it, but we find cool things.
We're doing two with a different animal, so now we should be friends.
Yes, we're friends. Yes. Should I be scared?
Yeah, no, it's fine. So you have all these different SNPs between the genotype, mitochondrial genotype. Do you know which one and which complex? Maybe they're like a pattern. Maybe some mitochondrial genome are more like different into the first one, and that can affect the type of trait.
That's actually
Another type of trait, maybe more
Yeah.
fitness, this one more activity.
Yeah. So we have that data. I haven't looked at it in depth, but I think that's a really good
And I think
thing.
you have like a pattern you could express
Yep.
from like just which complex are being modified.
Yeah, definitely. Yeah. I think we've got all that data, so I think it's just a question of compiling it in that way. Thank you. Yes.
The sterile combination, is there any similarity with the Brazil one, which is also sterile, in terms of that's not
It's a different SNP. It's a completely different SNP, so it's a completely different mechanism. And we've been working a lot more with this FA lethal, just because it's easier to work with lethals than steriles, but that's on the cards for next. Yeah. Yes? Oh, no, can, okay.
A very
Quick one.
I was curious if there are any intuitive or like theoretical explanations for why the mother's birth migration is successful. Is there an intuitive reason or intuitive counter reason for why it doesn't?
For why it doesn't work.
I preface it, but I'm not from the light industry.
Yeah, no. So, I mean, the theory says that it should work, and I guess the question is why haven't males gone extinct? Right? Why, yeah. So the theory predicts that the nuclear genome will compensate for a lot of these male harming mutations. And there's a couple of papers that I've found in Drosophila OXPHOS genes that have been duplicated, and these duplicates have testis-specific expression. And we think that these testis-specific mitochondrial genes in the nucleus are compensating for mother's curse mitochondria. Sorry. Coffee? Thank you. Thank you. Thank you.
Thank you very much, everyone, for coming, and thanks especially to Andrea and Sophie for giving me the opportunity to tell you a little bit about my research, our lab's research on this unusual system of mitochondrial DNA inheritance in bivalves. And in particular, I'm going to focus in on what happens to this unusual system of mitochondrial DNA inheritance, the genomes associated with it, when freshwater mussels make the transition from having separate sexes to being hermaphroditic. And these are some of the mussels that we're currently working on that we've collected in Costa Rica. So a couple of native species, and tragically this is an introduced species, an invasive species that was brought in on aquaculture fish from Taiwan. So as I think it was Georgina talked about this morning, and this audience will appreciate that in animals, the paradigm is that mitochondria are strictly transmitted maternally. And the only exception to that are some bivalve mollusks. And this includes some marine mussels, some freshwater mussels, and some marine clams that do this. And again, Georgina nicely summarized how the system works. So males, as adults, are heteroplasmic. So they somehow take mom's mitochondrial DNA and make it predominant in their somatic tissues, and dad's mitochondrial DNA comes to dominate in the germline and in the sperm. So the sperm here have dad's mitochondrial DNA. Adult females are homoplasmic, typically, and so they only put mom's mitochondrial DNA in here. And so a fertilized egg can either become male, and somehow the male contribution becomes sequestered and makes its way into the germline. And based on work that we did 30 years ago at Dow, in the first couple of days, the male type disappears in embryos that are going to become female. So we end up with two sex-associated lineages, a male lineage and a female lineage. And I'm going to be talking about freshwater mussels, so I'll tell you a little bit about the life cycle of freshwater mussels. So typically they're dioecious or gonochoric. Those are synonyms for having separate sexes. And so the males broadcast their sperm out into the water column. The females draw it in and they fertilize their eggs. And then those embryos mature a little bit on modified gill tissue. And then the females will shoot their parasitic Glaucidia larvae out into the water column, and they ideally clamp onto the gills of a host fish. And then they'll mature there for a while and drop off and complete their life cycle. And this is an example of a Lampsillus mussel. They have an absolutely fascinating adaptation in the females. So this is not a fish. This is an extension of the mantle tissue of the female, and she'll actually flick that tissue. It looks like a fish. A predator will come in, take a nibble, and mom will shoot her larva into the gills of the predator. Sometimes it looks like a little fish, sometimes like a little worm, sometimes like a crustacean. It's not pervasive in the freshwater mussels, but it's pretty widespread in some groups anyway. I don't have to explain too much about mitochondria to this group. Key point here is that typically animal mitochondrial genomes, which are really small and quite efficient, code for 13 protein coding genes. But in the freshwater mussels, there are unique genes. There's a unique gene in the female transmitted genome, and there's a unique gene in the male transmitted genome that don't have any obvious homology with any other genes and don't appear to be associated with making ATP. So here again on your left we have a female genome showing this F open reading frame, so it codes for amino acids. And in the male genome, again, we have what we're calling the M-type open reading frame, or MORF. And so from some work that Sophie did a number of years ago, we first hypothesized that these may be associated somehow with sexual development. And now that Sophie's continuing to look at this, and Justin Havard is as well in different contexts. So back to hermaphroditism. This is a phylogeny of some freshwater mussels. You don't need to look at the details here. The black species shown in black here, are ones with separate sexes, and the ones in orange are hermaphrodites. So the point is that hermaphroditism has evolved independently a number of times. It doesn't seem to be a long-term evolutionarily stable strategy. They're all fairly shallow branches, but nonetheless, it's likely an adaptation to low-density environments and temporarily adaptive. But a key thing is that the hermaphroditic species that we've looked at here have all lost the M genome completely. So they only have an F-type, but a modified F-type genome. But because hermaphroditism has evolved independently multiple times, each case lets us look at the relationship between the transition from dioiche to hermaphroditism and its impact on these ORF genes. I don't have time to go into the details, but we think that in the freshwater mussels, the male and female lineages have been divergent for over 200 million years. We actually think they originated through one of those role reversal events that Georgina talked about this morning. So we think DUI is even older than that. But so these genomes are very, very divergent, and they've hung on to these ORF genes or had these ORF genes for a very, very long time. And so there's 40-50% sequence divergence between the male and female genomes, and it's incredible that they're still functioning against the same nuclear background. It's really quite amazing in itself. And in the hermaphroditic species, so if we look at a closely related hermaphrodite, it's relatively closely related to the F-ORF. We refer to this as a hermaphroditic genome, and the ORF gene there is an H-ORF, but it's really quite closely related overall to the female genome. But the ORF gene evolves faster, as we'll see, than other mitochondrial genes. So here's an alignment of the amino acid sequences for F-ORFs from a couple of dioecious species, and you can see even though there's an indel in there, there's a large degree of similarity between the amino acids of the two dioecious F-ORFs. And we throw in an H-ORF, and it has evolved wildly, but it hasn't disappeared, and in none of the species that we looked at has it disappeared completely. So it's hanging on to it. This ORF is still doing something. And if we look now at the hydrophobicity profiles, and of course that's what gives a string of amino acids its biochemical properties, whether it's embedded in a membrane or dangling into the cytoplasm or something like that. So the dioecious species have similar profiles, but if we look at the profile of the H-ORF, it's wildly different. It's much longer. There are these repeated regions and so on. So something interesting is going on there. So the problem is that all these H-ORF versus F-ORF comparisons that we have are relying on genomes that are quite divergent. The hermaphrodites that we're looking at are relatively divergent. And so we'd like to detect some populations of freshwater mussels that are in the process of transitioning from having separate sexes to being hermaphrodite or have recently made that transition. And a few years ago, we did a case study looking at a Canadian species, Spiganodon, and tried to develop a method to look for molecular signatures that would, give us some insight into cryptic hermaphroditism. And the key to this process was using this program AlignMe, which quantifies divergence in a string of amino acids based on their hydrophobicity profile. So AlignMe lines them up and then gives you a measure of the divergence essentially in the hydrophobicity profiles. And so the dioecious species have quite similar profiles. The hermaphrodite on the right compared to a dioecious species is quite different. So we're interested in the ORF divergence, but then to kind of look at a background baseline amount of divergence between the genomes we're looking at, we used COX-1, the DNA barcoding gene, as a control essentially of divergence. And then we plotted ORF divergence, measured using AlignMe, against COX-1 divergence. And so we did this for a number of dioecious and hermaphroditic species, and here are just some of the hermaphroditic species that we were looking at. And so again, we calculated the divergence for hydrophobicity using AlignMe, and then we also calculated COX-1 divergence, and then we plotted those values. And so these points in orange are the ORF versus COX-1 divergence values for all the dioecious species. And these are the AlignMe divergence values plotted against COX-1 for the hermaphroditic species. So you can see that they're all really quite a bit different. But so now again, what we want to do, going out into nature, sampling new species, and trying to find some that might possibly be in the process of transitioning or have relatively recently transitioned. And so a few years ago, I was fortunate enough to go to Costa Rica and do a little bit of field work, and we sampled a couple local native species. And this is one. It's Nephronius tempesquensis, and we collected it from an aquaculture operation in the Guanacaste region in Costa Rica. We only got 6 samples, and then we did some PCR using M-specific genome markers and found that 2 of the 6 did show evidence of an M genome, so they're putatively male, and 4 didn't, so those are putative females. So then we sequenced COX-1 and some other genes and did some phylogenetic work, and we got a paper submitted on the phylogenetic work. But we also sequenced the ORF gene to look at how divergent this ORF gene might be. And in following up on this, we actually, after the fact, found this unpublished PhD thesis from 40 years ago that suggested that this species, Nephronius tempesquensis, from the same watershed where we collected our sample, but not exactly the same location, had some evidence of a low proportion of hermaphroditic individuals. So it's possible that there are some hermaphrodites in our sample. So we compared the ORF sequence for Nephronius with the closest relevant we could find, which is Papanius. And so here's the overlap of those hydrophobicity profiles. And here's where Nephronius actually falls out for its ORF divergence compared to COX-1 divergence. And so these were our standard dioecious species. These were our known hermaphroditic species. It's kind of on the edge. So maybe there's something interesting there. It's a little bit encouraging that maybe this is a population that's transitioning or has recently transitioned. So we want to expand our sampling and go back actually to the precise location in the Rio Tempisque watershed where Dr. Amador Barricol found some hermaphrodites 40 years ago, see if we can get some hermaphrodites. And ideally, we should do some histological examinations to determine if they are, in fact, hermaphroditic. And maybe we'll find evidence of a mixed population of males, females, and hermaphrodites. And so I want to thank my co-authors for this work, especially my Costa Rican colleagues who couldn't have done it without their help. So thanks very much, and I'll happily answer any questions.
Thank you. Talking about incompatibilities, I think my talk is a bit incompatible with the whole thing today because I'm a physicist. In fact, I work with models of speciation, and when I started to work with this, I have this model for evolution of a species under mitonuclear selection. And then I came up with this problem of nuclear compensatory evolution, so I'm going to show here today my last results and maybe have some feedback from the people who really work with this. Yeah? Oh, sorry. Okay. So all people here have been talking today about the mitochondrial selection and fitness and how these things are entangled and how this is involved in the species evolution. So just to summarize what was my view as a physicist when I first saw this, is that cellular respiration, in fact, depends on both genomes, the mitochondrial genome and nuclear genome, and the so-called genetic mitonuclear interaction. What this has to do with species is that when you have populations that have these co-adapted sets of genomes, but for some reason you have a subdivision of the population, geographic subdivision, they will evolve different co-adapted sets. So in some point when they are put back together, you see that you have these incompatibilities. And this is supposed to be a first step to a process of speciation. Especially because these incompatibilities they have impact on fitness, that what you have heard today, how the various forms of impacting fitness because of mitonuclear incompatibilities. One thing, additional problem related to mitochondria is that because of the typical properties of mitochondrial DNA being highly mutable and inherited by the mother, there is this, it's more prone to accumulate deleterious mutations. Nature itself took care of it in several levels of biological levels. We have purifying selection, so since from cells to tissues to individuals and populations to try to filter these deleterious mutations, but somehow they still sometimes they can go through. So there is a hypothesis that somehow this mitonuclear interaction can lead with this deleterious mitochondrial mutations in the sense that when you have a mitochondria that has a mutation that has harmful, this somehow will drive the compensatory mutation in the nucleus to correct it. This is something that some people measure, but is debatable, and especially when the kind of the tools that you have to have evidence of this compensatory evolution. So that's why I was interested in trying to model this and try to understand what factors influence the occurrence of the compensatory evolution. So, as a physicist, my model is super simple. So it's very coarse-grained simplification of the machinery that we are talking about today. I work with this individual-based model in the sense that I have simulations with each individual is a point in space, and I have two sequences of each site can be translated to a gene that can be 0 or 1. So I have the nuclear DNA and the mitochondrial DNA for each individual. In the beginning, all individuals are identical, and then I have some rules for the evolution of this population such that with time we can see some groups break, and then we can see species evolving. So what are these rules? When I have individuals, they're gonna mate, they only will mate if they are especially close, so I have spatial restriction to mating, and they have to be genetically close, in the meaning that their nuclear DNAs has to be very similar. An additional ingredient that I put in my model is that we have then the mitonuclear selection. So each individual, I compare the two sequences side by side, and I count how many sites are different, and then I call this the mitonuclear distance. It's a very phenomenological form of modeling what we have seen here about the compatibility of the proteins that are encoded by the two genomes. So as I do this measurement of fitness, and this gives the likelihood of the individual to reproduce. So individuals that have a higher mitonuclear distance, they are less likely to reproduce. So I do the dynamics of the population. We also have mutations that happen in the moment of the offsprings are generated. And one important thing is in our model, the mutations per se are neutral in the sense that 0 or 1 is not, there's no difference between them. The only point of selection here is that when you have the difference in the respective site of the nuclear mitochondrial DNA, then you have a loss of fitness. With this model, I starting from fully compatible populations. I studied some signatures in speciation. So in previous works, I studied what this affects in species lifetimes, abundances, speciation rates, extinction rates, hybridization. But now I want to look what happens when I start from populations that are not fully compatible. So remember that all individuals are the same, they're kind of clonal in the beginning of the simulation, but all of them will have a part of the mitochondrial DNA incompatible. So I have two portions here, one portion that's incompatible and a portion that is compatible. And then I can check how each portion is gonna evolve. I can check the substitutions in each site. We kind of make an analogy with mitochondrial integration, in the sense that we have some populations that you have this invasion of a mitochondrial DNA that's incompatible, and then we see what happens in evolution, like how population can survive even in the face of incompatibilities. So first of all, we look at the effect of the mitochondrial mutation rate, because one thing that is expected is that increasing the mitochondrial mutation rate would increase the likelihood of the nuclear compensation. So once you have a lot of mutation in the mitochondria, you have more, the nucleus would be more selected to compensate those mutations. And we see some intuitive things. So in the first row, we have the substitutions in the nuclear DNA, and in the bottom, in the mitochondrial DNA. Here's the portion that starts with the both compatible parts. The black line here is a reference for us of non-interacting nuclear genes. So in fact, when you have the selection, the substitutions in the nuclear genes tend to be decreased because the systems want to keep the compatibility. But as you increase the mitochondrial mutation rate, this goes up. This occurs because, of course, the mitochondria starts to have more substitutions. And at some point, if the mitochondrial mutation rate is too high, this is even the substitutions even go past the non-interacting sites. What was non-intuitive for us is that the sites that start with incompatibilities, in fact, they decrease the substitution rate as we increase the mutation rate. Maybe this is a problem in our model, that what happens is if you increase mutation rate, in fact, the mitochondria can rectify this incompatibility. Or maybe this is something that we have to look for in nature, that there is somehow the mitochondria, because it has a high mutation rate, could rectify these incompatibilities. So when you look like it's the same data, but just looking to the nucleus, what we call the compensatory evolution is the blue curves, are the curves of the sites that became incompatible. They are always above the level of non-interacting level of the non-interacting genes, but you see that increasing mutation decrease this substitution rate. And in fact, the sites that start this compatible, they have more substitutions. So it's kind of the counter effects, is like how the nucleus behavior depends on how they were in the beginning, the starting states. For competitions, we also looked in the selection strand. In the match genes, this was expected because selection will try to tend to maintain the compatibility. So you decrease the substitution in both genomes. And in the nuclear genome, if it starts incompatible, selection will increase the substitutions. For the mitochondrial genome, it's kind of a mess. If you have kind of intermediary selection, you have the substitutions are higher than the case with no selection. But if selection is very strong, it also tends to, at some point, impaired of accumulating too many mutations. So it's not also linear. And in the end, we also looked into how many incompatible sites we have. Sorry. So this is more kind of expected, like in the matched sites, we don't see any big effect. But in the nuclear sites, as if you have more incompatibilities, you have more substitutions, and the mitochondria is the opposed. So if you summarize, the non-compensatory evolution is promoted by strong selection, but somehow has different effects because of the mitochondrial mutation rate. I had something to say about speciation too, but just to summarize, we can compare the situation without incompatibilities and with compatibilities, when you have incompatibilities, and what we see is that the number of species is not affected, but speciation tends to occur earlier. So somehow incompatibilities are driving speciation. So sorry, I open my time, but the idea here is to motivate the study of models. Of course, your models always have to talk to reality to understand if you're really obtaining results that make sense. But I understand that even like in simplified models, you can understand how factors influence the outcomes of this process. So thank you.
Great. My name is Erik Iverson. I'm from the University of Texas at Austin, and I'm going to talk about ecological speciation and mitonuclear co-adaptation on elevation gradients. And I want to thank my co-authors, some of whom are in the room with us here. So I'm going to be talking today about a very common ecological pattern, which is that as you move up in elevation, especially on tropical mountains, you often see closely related species replacing each other with elevation in a way that looks like they are adapted to progressively higher altitudes, to the rigors of cold and low oxygen that come with living at higher altitude. And even where one species covers a range of altitudes, we often see that there might be some specialization in the mitochondrial genome. So an example is this rufous-collared sparrow from the Andes, where the nuclear genome, which is represented by these rectangular chromosomes, is undifferentiated across elevations. But the mitochondrial genome, which is represented by a circle in a convention that we're going to use in this talk, is actually showing differentiation across all those altitudes. And one reason that you might see this pattern is that in a variety of taxa, including in humans, there has been extensive work to characterize mitochondrial substitutions that actually appear to be adaptive for either low oxygen availability or cold temperatures and help animals live at high elevation. Now, I'm going to skip over all the functional stuff about how mitochondria work, because you already know that. But essentially, what you need to know is that there are a variety of reasons why we might expect mitochondrial genes to have a role in adaptation to different climates, whether that's about the diet that these animals are encountering, the availability of oxygen in the environment, or UV radiation's impact, and the temperature and the rigors of living as an ectotherm or an endotherm. All of these things can play a role in adaptation through mitochondrial function. What you also already know about mitochondria is that the main enzyme complexes they have are chimeric, so they contain parts that are encoded both by the mitochondrial genome and the nuclear genome. So those components encoded by the mitochondrial genome are in green here, and those by the nuclear genome in yellow, and everything that's orange and red is where these protein residues are interacting. And that is leading to tight coevolution between these genomes, where there may be compensation for the change in one genome by the other one to maintain a fit between these proteins. One of the consequences of this is that you have a gene by environment interaction that's going to shape the response of these organisms to the climate and can also contribute to reproductive isolation between organisms. So what we're looking at here in the case of these seed beetles is a study where on different nuclear genetic backgrounds on the x-axis, you then have the different lines going across our different mitochondrial genetic strains, and essentially every combination of nuclear and mitochondrial is showing a different metabolic rate. And also we are changing temperature between the top panel and the bottom panel, and you can see that all those relationships between those two genomes and metabolic rate are then switched based on what the temperature is also. So you need to account for the interaction of these two genomes and the temperature to understand the metabolic rate of these organisms. Now, there's a nice study that we did in collaboration with the Schumer Lab at Stanford University recently that kind of highlights the importance of these mitonuclear interactions and how they can enforce reproductive isolation between species. And that's by looking at these two species of swordtail fishes from the mountains of eastern Mexico and the hybrids that they form at intermediate elevations where they come into contact with one another. And so if you look at a big admixed population of these hybrids, you see that there are certain places in the genome where if you have the mitochondrial haplotype of one species, you never have homozygous nuclear ancestry from the other species. And we can identify that one of these important interacting genes is actually part of complex 1 and use protein modeling to determine which residues are interacting there and causing this lethal mitonuclear incompatibility between species. And then using mitochondrial respiration analysis, we have been able to show that the hybrids between these two species, shown in the left panel there, they actually have a compromised function in complex 1, so they show lower mitochondrial respiration in complex 1, but not in other measures of mitochondrial function. So that's good evidence for us that what's going on here is basically because evolution has gone down two different paths in these two different species, when you bring the mitochondria from one species to work with the nuclear genes of another species, you have this reduction in function here. And we don't know if this is because these two species have mitochondrial adaptations to different water temperature or whether this is because of genetic drift. That's something that I'm looking at in another project that I'm not going to have time to talk about today. But one of the implications of this work for me has been that, if mitochondria are playing this important role in adapting to the environment, then we might ask whether we can use our understanding of mitochondrial adaptation for a conservation purpose. A lot of people are talking these days about facilitated evolution, helping species adapt to climate change maybe by giving them the genetic variation that they need to survive in a warming world. And so I put forward this idea that we could actually move mitochondrial haplotypes between species in a way that mimics a natural process whereby mitochondrial haplotypes introgress, and it often looks like it's adaptive and related to climate. But you would need to account for these mitonuclear incompatibilities, right? So if you're moving a mitochondrial genome from one species into another, you're going to cause incompatibility with the nuclear genes that are there. So the solution for dealing with this that I put forward in this paper would be on the pathway along the bottom, you would essentially combine both mitochondrial replacement with editing of those key nuclear genes that interact with the mitochondria in a way that would allow you to overcome those mitonuclear incompatibilities and move mitochondria between species in a way that might be adaptive to do so. Now, why might you do this? Well, if we're thinking about mountains again and the fact that closely related species replace each other with elevation, we may have a scenario where everybody is moving up in elevation, and there are cases in which species are going to go extinct at the top of a mountain, or they may be unable to move up in elevation due to a barrier of low oxygen content or something. And so if we're able to move mitochondrial haplotypes that are very adaptive from closely related species that live in other climate space, maybe climate space that's more reflective of what these species are going to encounter in the future, then we may be able to help these species either persist on a mountain as it gets warmer or move up in elevation in a way that they wouldn't have been able to do. So the next thing I'm going to talk about is a test for whether there are actually functional adaptive differences in mitochondria across elevations. And so we're going to work with these birds called tapaculos that live all across the Andes Mountains. And you can see on this phylogeny, they're coded by the elevation at which these species live. So they inhabit a really diverse breadth of climatic niches. So I'm looking at 4 species that replace each other on the same elevation gradient in southeastern Peru. These species can be found by just making this transect up the Andes Mountains in this region of Peru. They have these differences in their calls that seem to suggest that they are reproductively isolated and good species. And so what we did was we went down to Peru and we measured the basal metabolic rate of these birds, and also we measured their mitochondrial function. And in particular, I was interested in a measure of mitochondrial heat production that could be important for endotherms adapting to different climates. And so I worked with this really amazing team down in Peru to catch these little birds and do this work. And so when we measure the metabolic rate of one of these birds, we're getting a trace like this where you have all these measurements of oxygen consumption as we lower the temperature, moving from left to right. And we use AIC to tell us how many breakpoints we should put in this graph. And one of our important measures of metabolic adaptation is where the lower critical temperature is, which is essentially where on the graph, whichever one has the lowest AIC, where's that breakpoint where those species seem to increase their metabolism as it gets colder. And what we see is by looking across these 4 species from left to right, it's the low elevation ones to the high elevation ones, that point at which those birds start to increase their metabolic rate dramatically goes down as you move up in elevation. So this looks like a measure of thermal adaptation, whereby the ones from higher on the mountain, you can get them colder before they need to dramatically increase their metabolic rate. And so we also dragged this mitochondrial respirometer down to Peru with us. Here's an undergrad using this in the field there, and what I'm going to show is one measurement called leak respiration, which is essentially how leaky is that mitochondrial membrane, how much of that energy input is being diverted to basically maintain the heat of the body rather than being used for useful energy. And what we see in both the brain and the liver is no signature that we can determine across elevation. So if there are metabolic differences between these birds, it doesn't look like it's anything as obvious as just having differences in leak respiration. But there's a bunch of other mitochondrial measurements we could talk about. So just to put those next to each other again, the mitochondrial leak respiration looks like it's not very different between species, but our whole organism measurement of metabolic rate is showing some potentially adaptive differences. Final thing I'll talk about is a study to look across a much wider range of organisms and ask about the roles of both adaptation and neutral processes in driving differentiation in the mitochondrial genome. And so we're looking at mitochondrial genomes from about 150 different high elevation species and their low elevation relatives in this phylogenetic context where we want to know about both the DNDS value and the K value, which is a measure of relaxed selection. And in this framework, if you measure DNDS and K, you can get a good sense of what kind of selection is going on. Are you actually having positive selection? Are you having relaxed selection? Is that relaxed selection the relaxation of positive selection or the relaxation of purifying selection? And we put out a paper last year that kind of explains this framework for thinking about adaptation. And so 150 of these comparisons where we take a high elevation species on the left there, compare it to a low elevation sister species and polarize that with a low elevation outgroup so we can ask about DNDS and K on those high elevation branches. And so this is a preprint now that you can look up if you're interested. What we see is essentially in this framework, most groups of organisms, most groups of animals that we looked at, they show a slightly higher DNDS value among those high elevation species. But when we ask about a few other measurements, we see that we took a bunch of ecological data into account also, like range sizes and latitudes of these ranges. And we see that the range size plays a big role seemingly in driving this trend also. So if you have a smaller relative range as a high elevation species, you're also more likely to have a higher DNDS value. Those small ranges are also associated with being higher up in elevation and with having a more relaxed K value. So what it looks like is going on maybe is that high elevation species are living in smaller ranges, lower effective population sizes that might be driving higher genetic drift and thus explaining this pattern of higher DNDS ratios. And we looked at this also in this phylogenetic framework, sort of family by family, where we see as you move across elevation from left to right, that DNDS values tend to get higher in most of these groups and K values tend to get lower. So once again, that's telling us that there is probably relaxed purifying selection going on. And so again, that relationship I explained may be that high elevation species have smaller ranges. They're going to tend to live at lower effective population sizes, leading to drift. And that could be explaining this widespread signal of higher DNDS values. So we're there on that chart, high DNDS, low K, being relaxed purifying selection. So to summarize all of this, I want to put forward a model of how mitochondrial and mitonuclear coevolution can kind of proceed in species that become isolated from one another. So as you move from left to right in this graph, imagine one population becoming split in two by a barrier and starting to accumulate differences in the mitochondrial genome and the nuclear genome. Now, some of those differences are going to be adaptations to a hot or a cold background environment. That's that colored background going on there. But some of them are also thermally neutral, so they may just be accumulating due to drift. But whatever the case, when those organisms come back into secondary contact, they're going to have two different forms of mitonuclear incompatibility potentially. One is incompatibilities between hot adapted and cold adapted genes, and another is just between differences that have nothing to do with climate and have just accumulated due to genetic drift. And so they're going to have reproductive isolation. They're going to shake out on that environmental gradient wherever competition allows them to. And we can also think about differences in effective population size as something that might be driving mitonuclear differences between these species. So those high elevation species might be having that lower effective population size. They're going to have a more rapid mitochondrial evolutionary rate, and differences in rate is something we saw in the swordtail work and in other work can have a big impact on driving genetic incompatibilities. When one species is evolving quickly and another is evolving more slowly, then that often leads to genetic incompatibilities. So in conclusion, both neutral and adaptive forces may drive evolutionary divergence between species at different elevations. Mitochondrial and mitonuclear differentiation, whether adaptive or neutral, can contribute to reproductive isolation via mitonuclear incompatibilities. And we can consider mitochondrial haplotypes and NMT genes as key loci in conservation genetics for their role in both adaptation and reproductive isolation. I know that was a lot. This was originally a Hamilton talk, so it was supposed to be like, oh, let's cover all of your dissertation work in one talk. So if you want to go more in depth with anything, please find me after the talk. I'd be happy to talk about it. I want to thank all the societies that put on this meeting. They've all supported me at one point or another. Thank our collaborators at Stanford, as well as the awesome field stations that let us do work there, and some of the other people who have contributed to this work. So thank you.